Hello to everyone. Welcome to geneXplain web seminars. This is actually our next web seminar on the topic of COVID-19 research. And we had two web seminars where Dr. Kell has shared with us how to analyze gene expression data in COVID-19 patients versus healthy control. We had also two web seminars by Professor Paroikov devoted to the cheminformatics side on research on this topic and identification of biological activities of chemical compounds. And today our focus is on the approaches how to analyze regulatory SNPs associated with COVID-19 disease. Before we start, I just need to mention that there is Q&A box in this interface, and you are very welcome to put all your questions in this Q&A box during the whole presentation. And after presentation by Dr. Kell, he will definitely take time and answer all your questions. We will try to answer all your questions. Hope we had time enough, and we aim for presentation by 30-40 minutes, and then we answer all your questions. So now, Alexander, please take over.
Thank you very much, Olga. So, hello, everybody. So let's start the seminar today. I will share my screen, and then we are going to start this presentation. Basically, the plan is first to show you some introductory slides, and then we'll proceed with the online demo, say 15 minutes of the slides, and then whatever time we need, maybe around 15-20 minutes, we'll do the demo. So today, as Olga said, we are talking about SNPs, SNPs, and those ones which are non-coding, so regulatory ones. And as you know, it's actually very surprising sometimes, but 95% or even more of those SNPs which are actually associated with diseases of various types are not coding. So they are not located in any gene. And they are not actually changing the expression, they are not changing the structure of the protein. So instead, they are located between the genes or in the promoter of the gene, enhancer of the gene, and changing their expression. They are changing expression of the genes. Although, since we know more about the coding, of the proteins, most of the attention so far is put into those SNPs which are located in the proteins, and indeed they actually damaging the structure of the proteins. They are changing the structure, and they may influence a lot on the disease. But actually, we are losing 95% of the information when we're just focusing on the coding SNPs. That's why we want to change that. We want to look at the non-coding ones, and especially with a focus today on COVID-19 susceptibility. So we use data from this very nice website where several groups started to collect genome-wide association studies and made analysis of that data to learn and to determine genetic determinants of COVID-19 susceptibility. So basically, predisposition of the people to be ill or severe ill from this COVID infection. So we'll use this data today to demonstrate how we can analyze regulatory SNPs. So regulatory SNPs, as I said, they are located in the regulatory regions, basically between the genes, and they can do a lot. They can change many things in the DNA, but our focus today is on those SNPs which can change the binding sites for transcription factors. So they can change many things. For instance, they can change binding sites for microRNAs, or they can change stability of some complexes or anything. But majority of effects, as far as we know, is actually because of changes of the DNA binding properties of transcription factors. So for instance, here we have AP1 transcription factor, and in case if a certain mutation or certain variance is observed in a DNA place where this protein is binding, then the site can either be gained or lost. For instance, there are cases, known cases of variations or mutations which are actually disease causative, and they are located not in the genes. So, for instance, this change of A to C in this position of TCF1 gene, in the promoter of that gene, is known to be associated with diabetes. And when this A is changed to C, actually the site for COPE or DR1 is lost. So, and again, another case when this mutation in this gene and this variance, in fact, leads to a new gain of a new site. So in a T, when there is a T here, there is no site, and when the T is changed to C, or the variant is C, then the site can be observed here, and this change is known to be related to this particular disease. Actually, one change of position can, in the same time, kill one site and create another site. So like in this case. So all these cases are either predicted or known, but basically there's a lot of them in genome. And to do, the systematic search of those cases when we see the SNPs and we want to see how these SNPs are changing the potential binding sites in genome, we are using TRANSFAC database, the golden standard of the collection of predictive models for predicting binding sites for transcription factors. And currently, we have around, we actually have 7,600 different positional weight matrices for vertebrates, vertebrate factors. Just top of that list is presented here. So you see basically the name of the transcription factor family, or individual transcription factor, which is binding to the motif, which is presented here by this logo. And we have 7,600 of these positional weight matrices, which are then can be used basically in the following way. So if we have a matrix like this in TRANSFAC, then, and as you know, this matrix was built most of the cases by collecting known sites, for instance, for one transcription factor, say E2F1, and then the matrix is calculated based on the frequency of nucleotides which were observed in the particular positions of those sites. And when the letter is high, that means that these frequencies of in this position always say C or T was observed in the real sites for E2F1. And now, when we have this matrix prepared of some known sites, we can apply it to a new sequence, to a new promoter sequence, or a new genomic sequence, which we believe is involved in regulation of a gene. And then we can actually calculate a score, which means how well this position is actually fit to this consensus, or more exactly to this positional weight matrix. But what happens if a particular position is in this place is changed? So we see that in one case it was T, and then it's changed to G. So then immediately, using the same matrix, we can calculate a new score of that place, of that position. And then in case if we see the drop of the score, for instance, the new score after this change is much lower than the original score, we can say that this site was lost. And in other way, if this Q is becoming bigger and much bigger and bigger, higher than a particular threshold, we can say that this site is gained. So it appears in this place. And in fact, we know that this binding of transcription factors to the places in genome are not exact. So we should forget this concept of exact fit. In fact, they are binding very proximate, and it leads to the problem that actually the same place of genome can be bound by different transcription factors. We can predict many different sites on the same place, and also we know experimentally that indeed in different situations, different conditions in the cells, this place in genome can be targeted by different transcription factors. So, and if we observe a mutation in that place, it can influence not just one site, but several of the binding of different sites. Moreover, this is actually observed in vivo. Here you see example of experiment which was done in a cell line, and colon cancer cell line actually, where an enhancer of a MYC gene, there is one SNP, and one variant of a change of a nucleotide. And in that line, which carries this SNP, the experimentally, we observed a lot of binding for many transcription factors to that place. Actually, the whole place is rearranged. So in case of this SNP, not only the binding sites which are immediately affected by this SNP are affected, but also the region around is getting rearranged. And why is that? Because we know that transcription factors, they are not actually binding alone. They are binding in accomplices, so-called composite modules. So when the binding sites for a central transcription factor, say in the center, say this is a factor RAF and it's binding to this place, but in nearby there are other binding sites for other factors, and only when these 3 or 4 or 5 transcription factors coming together and binding in synergistic manner to this region, then the particular change of expression is observed in that gene. So, and we come with this concept of fuzzy puzzle. Basically, since this binding is not very exact, then one mutation in place can distort the whole region, and the complex of factors can be substituted by another complex of factors, which would drastically change the expression of the gene. So, and to analyze that, we come up with an idea to apply our composite module analyst, the algorithm which searches for the composition of binding sites, not just at the place where the mutation is happening, but at the region around this place. So that's why we can not only look at the transcription factor binding sites, which is immediately affected by this change of a nucleotide, but we can also look at how the whole region is rearranged after the mutation. And here we have this algorithm, so I will not go too much into details, saying just this algorithm is actually applying genetic algorithms. It searches for the combination of transcription factor binding sites in a given region, and at the end it gives such output where it come up is a composition of transcription factor matrices, which you see here, and then each region we are looking at is then searched by these matrices, and then the algorithm finds clusters of them. Basically, you see these gray areas where the region of genome near the binding, near this change of nucleotide, near the SNP, is actually enriched by a cluster of those transcription factor binding sites. And actually, you see that if we compare the concentration of those clusters, those binding sites, in the regions with mutation, and compare them with other SNPs not related to this disease, for instance, then we see clearly separation. So these regions around the SNPs related to a particular disease, they are saturated, they are enriched by many sites for a composition of transcription factors. And the next step after we find these binding sites, we could ask a question how these transcription factors are regulated, and we are looking for master regulators in the signal transduction pathway. I will show you a bit of that today, but still our focus mainly today on the transcription factor binding story, just to let you know that how we actually search for master regulators. we look at our TRANSFAC database, a database on signal transduction reactions, and we are looking for basically searches in this network for master regulators, which can lead to activation of several transcription factors in the cell. We also check if there is a certain change in or potential change in this setup. We call it working pathways, when the master regulator, which must regulate these genes in the standard conditions, in the disease conditions, is changed by a new master regulator because of multiple feedback loops. And I will touch that a little bit in my presentation today. And we see then these disease conditions, for instance, when we have inflammation conditions in the cells, they are characterized by many feedback loops when the activated master regulator leads to the activation of the genes which encode the master regulator and which are encoding the whole components of signal transduction pathway leading to those genes. So basically those red stars are indicating the SNPs, and in the disease conditions, those SNPs can lead to the feedback loops, which may determine the higher susceptibility to a particular disease. So today I will look at this data from this COVID-19 website, and there are several comparisons which were done here. So basically, a lot of people, a lot of patients were genotyped using microarrays, and then there was a comparison already done in this tool. And in this comparison, for instance, when they compare hospitalized COVID-19 patients, they compare them to the total population. So basically, they had 3,199 hospitalized COVID patients in that cohort, and then they compared to the population, which has around 900,000 genomes. And then clearly you see on this slide that here you see the chromosomes starting from chromosome number 1 to chromosome number 23. And then the peaks correspond to those SNPs actually, or combinations or several SNPs, which are actually getting very high p-value, well, very low p-value, very significant SNPs, which means these SNPs in that regions, they are associated with people who were hospitalized with COVID-19. Another comparison was done by comparing very severe respiratory confirmed COVID-19. So among those who were hospitalized, 536 were very severe. And again, that was compared to the overall population. And although this study is has not so many cases, so that means it was very difficult to get significant SNPs, but still some of them get through this cutoff, and we can get these SNPs for our analysis. Okay, so now we are going for the demo. Let me now change to our tool. So today we are going to use the tool which is called Genome Enhancer. So it's one of our tools which was just recently developed, and this is a tool which can analyze many omics data, and I already demonstrated how to analyze gene expression data from COVID and other diseases. And today I'm focusing, as I said, on genome-wide associated data, on SNP data. So what we are going to load in this system is a list of SNPs. Here it is in a file, which is a VCF format. And this is a SNPs. These are SNPs which were found in the hospitalized COVID-19 compared to the overall population. So, and the p-value cutoff was in actually 10-4. So, and you see a number of SNPs here, quite high number of SNPs, but of course we put a very low cutoff here, not to miss any, regulation. And I will touch that cutoff issue a bit later. Okay, the project was already created, so we would open it. And actually, to load the data, you have to drag and drop this file. So let me just find this file. It's here. So we drag and drop it here. And so we, by the way, you see before we also tried with another cutoff, 10-5, so a smaller number of SNPs. Here we can take bigger numbers. So here we have to say which kind of which genome build is used to for the position of those SNPs. So it was the latest genome build, so we can import it. And then we press next. And then basically what we do, we take this data, which we just imported, it's marked here, and drag it here in this gray box it's one is already there, so we want to use this one, so we drag it here, and then we press next, and here we can make actually a new category for our analysis. So as you see here, we used already a category for those SNPs which are with a cutoff 10-5. We can create a new category with another set of SNPs. Let me call it, new, for instance. And then we drag and drop it here. And then we press next. And that's the only thing which we have to specify is, in fact, we have to say what kind of category we are analyzing now. And we just use one condition. So if in case we would have several conditions, we could compare them, but here we have just one, so list of SNPs. And then we have to specify the disease we are interested in. So here we, of course, are interested in virus diseases, and here we have a coronavirus infection. We can select the general virus diseases or specifically RNA virus infection. We can also specify a bit more broader set of diseases, saying we are interested in all diseases which cause severe acute respiratory syndrome. For instance, in order to see, basically this selection of diseases will help us to focus on the drugs later on. So that's you will see in the report. So basically that's what is needed. So then what is next is just to start the analysis. And then this analysis will run for several hours. So it's actually because of making the genetic algorithm and estimating the probabilities. So it takes some time, but we will look at the already ready result. So when I analyze here the set of SNPs which are with a cutoff 10-5, so a smaller subset. So we click here on full story, and after analyzing all this data, the system generates the full report. It actually finds the most important targets, let's call it. So it finds those master regulators, you remember from my presentation, which can influence on expression of their genes through those positions in genome which carry those SNPs. And in our case here, the system detects interferon receptor and this gene. So basically, we'll look at these two genes later on, what they are doing and what are their interest in behavior and features. It's actually a very interesting story at that. So and then what first it is doing is actually mapping all these SNPs on genome and checking whether those SNPs are actually hitting a particular genes whether they're hitting exons of those genes or introns. And you can see here on this diagram the top gene which carry 38 variants, SNPs, inside the body of the gene is interferon receptor 2. You see that the blue boxes are the exons, and the arcs are actually introns, and you see that most of the SNPs are located in the intron regions. So you see here other genes. For instance, this gene was also very interesting, chemokine receptor 6. That was the biggest peak, which I showed in the picture, in terms of probabilities, in terms of p-values. This gene is actually having a SNPs where is the highest link to the disease, to this, to the COVID hospitalization. But according to the number of variations which are covering the gene, and also actually the gene also bigger than this one, the interferon receptor here at the top. But then actually we get all these genes, and then we want to ask several questions: how these genes are related to each other, what they are doing, and so the functional analysis in terms of gene ontology is done. Then basically we check how those genes are mapped to the canonical pathways, and in fact you see several pathways like interferon-related pathways and TNF-related pathways, and also some pathways related to metabolism of thyroid hormones affected by those genes. So, but still, you don't see the full picture. Basically, many functions are affected by those SNPs, but which one is the biggest one? That's the question. And in fact, if you map those genes to various known biomarkers of various diseases, we can see interesting thing that in fact many of them are mapping to known biomarkers of infections, for example, bacterial infections, right? Or mycosis, so other type of infections. But at the top, the majority of genes actually can be mapped to carcinoma or neural diseases. So actually those genes are of various influence. And of course, we are looking at a very crude mixture. So these SNPs are associated with hospitalization of COVID-19, and what kind of process these SNPs are working with is absolutely not clear. Whether it's related to infection, whether it's related basically to the processes in the tissues which are related with a severe response of the body, or maybe it's related to some neurodegenerative disorders. Who knows? So that's all the crude mixture. But we now are going to focus on the biggest impact. And to find the biggest impact, we first of all look at the transcription factor binding sites, which are affected by those SNPs. And here is a little statistic. So you see those SNPs here again. And now we look only those SNPs which may change binding sites for transcription factors. They can either, by a SNP, the site could either appear, so we can either gain the site, or we can lose the site. And then we can count how many, among those all SNPs which we observe here, how many of them actually kill sites of particular transcription factor. And we can estimate the p-value. For instance, if in this case SOX9 sites were killed in 34 times, it appears to be significant compared to the random chance. So we can say that definitely those SNPs prefer to kill SOX9 site. And on another side, for instance, and we can look at the RAF1 sites, and this is a focus today. In this case, we got 13 certain sites, RAF sites, which appear here because of SNPs in alternative alleles, but it's happened to be also significant. So by random chance, it must be lower, much lower number of sites of RF factor could appear just by random chance. So this give us a particular focus on those transcription factors which may be influenced by those SNPs altogether. So what happens in those people who carry those SNPs in the regulatory regions, they often affecting sites for these transcription factors. And then this may lead to change of regulation of the corresponding genes. To even further, we can, as I said, we can look around those binding sites and around those SNPs, not just focusing on the particular region, a particular place, which nucleotide is changed by this SNP, but looking in the bigger region. And the bigger region means 1,000 nucleotide, 500 left from the SNP and 500 right from the SNP. So, or SNPs are around those, the region around those SNPs. And when those regions were looked and analyzed all together, it appeared that there were very clear signal, actually, which separate all those SNP regions, SNPs carrying regions, from any random region in genome, actually carrying other types of SNPs. In fact, as a negative set, we look here at the regions of genome which are located near other SNPs, not correlated with the COVID infection. And if you look at the transcription factors which were selected here, you see a very nice combination of OCT and non-OCT. These are known factors for which are related to the growth and to many processes in the cells, and also related to stemness, so-called stemness to stem cells. We see other factors which we will talk a little bit later. So at the end, the system builds such a diagram, and I will show it from bottom to top. So it finds these three top genes, interferon receptor, HDHC4, and RBL1, which carry in the upregulated or in the upstream regions or in introns of those genes. They carry sites for these transcription factors in clusters. And these transcription factors are in turn activated through this signal cascade. And at the top of that cascade, you see interferon receptor, HDAC4 protein, and P107, which is a protein encoded by this retinoblastoma ligand 1 gene. So basically, you see that this network explains how these genes are connected. So this regulation leads to potentially to the change of expression of that genes through the SNPs, and they in turn encode the proteins which are these receptors. So this creates these feedback loops, which may lead to the growth of the of the inflammation, or to the rapid increase of the processes involved here. Okay, so but today we want to instead of going further to look more towards drugs which may be associated with these drug targets, as we did before when we studied gene expression data. Here we are interested more in the details of the mechanism, how these genes are regulated through this SNP, and how they are regulated through the transcription factor binding sites which are affected by these SNPs. To go into more details, we have to switch here to a more focused type of our work. So here we use the Genome Enhancer view on our platform. Here we switch to the full platform view. You can see what now we can work here, how we can work here. Basically, with this platform view, we get more functions. So we get actually a lot of functions to analyze the data in details. We actually can also look at the results of this pipeline of the analysis, and look at the more details of those results. So we can go directly to this project and find our results. Open the project first. So we were analyzing this COVID data, and here the results are located in this subfolder. And here, these are the files, all the files which were generated by the system through the analysis. And the report itself is also here, so we can open it again and see. But first of all, we want to look in more details of those composite elements. So they are here. If we click on them, we can see actually the structure of that composite module. But now we look at a bit bigger module, extended it here. So on the way of analysis, it's first analyzed the extended model, and then it made a smaller one. But here we want to look at the all found sites. So you're looking now at the extended model. You can see it here. So again, you can see here the distribution, how this model around the binding, around the SNPs, these sites for these transcription factors are enriched in the regions around the SNPs. You can see each region here in more detail. So for instance, now we are focusing on a region which is located in the interferon receptor gene itself, because as we discussed, this region then creates this famous feedback loop. So the interferon receptor fires the signaling, which actually targeting the transcription factors, which are binding in the region of that gene, which in turn may influence expression of that gene itself. So, and we can have a look in more detail of that region by looking at the browser. You see those composition of binding sites, and among them you see this IRF1 binding site, and you see the OCT binding sites, and also SOCs. They are very, and non-OCT, OCT and non-OCT, those ones which were selected as most important. So you can also look at the more details in the genome browser view of that region. So if you open the genome browser view of that region, we can actually look in more details. So you see here this region, and we can zoom in to the level of nucleotides. So we can zoom very, very far away to the level of nucleotides. We can see, let me jump into this RAF site. So this RAF site is actually affected by those SNPs which we identified. So this SNP is identified by as associated with COVID hospitalization, and it's located in intron of RAF receptor. And as we can see, this this SNP, by changing C to actually T to C, so the reference allele is T, and its alternative allele is C. And when it's changed to C, it creates this RAF binding site. And you can see here on the statistic of that site, by the way, here is the consensus, and it changes exactly this G. So in fact, you see the site is like going not from left to right, but from right to left. So it's on the opposite strand. So changing T to C means changing A to G on the opposite strand. So in the reference allele, there was A here, and obviously no any RF site, but as soon as it changes to G, actually it is clear RF site with a high score, and the score difference, so because of the change, is 0.8. So actually it's appeared to be in a very conserved place in this site. But what was interesting actually to look at the what happening in this region in general. And we can zoom out a little bit, this region, and we can plot here some more information. For instance, we can plot here data from other databases. There's a database, a famous database, which collects EQTLs. So those, basically those SNPs, which are correlated with expression of genes. So this GTX database. And in that database, in different tissues, basically it gives the list of SNPs which are correlated with the expression of different genes. And we can take those SNPs and plot them here. For instance, I already plot the lung tract, so I can also plot, for instance, liver tract. I can plot here liver. And or in case of liver, there is no SNPs in that regions which would be associated with expression of interferon R receptor. But in case of lung, there is a lot of such SNPs associated with this expression of that gene, and they are located exactly in this region. So if you now look at this SNP, which we are focusing on, so you can see that it covers by many of these SNPs which are associated with expression of that gene, like this one. In addition, we can map here actually ChIP-seq data which we get from ENCODE project. So we can map here the data of ENCODE project of ChIP-seq, which was done for IRF transcription factors. And again, you see here that there are a number of ChIP-seq peaks which are located exactly at those SNPs. So this is a SNP which we are talking about, which is changing the IRF site, which you see here. And you see here this IRF4 ChIP-seq fragments. They are covering this SNP region. Also, as you remember, we found also other transcription factors like OCT1 and other OCT factors located nearby. And exactly, if you look at the fragments for the POL2, which is OCT family factors, they located. These peaks are located in the regions nearby. So as you can see, by adding more and more information here, we can look at particular SNPs and build our evidence that those SNPs are really influencing expression of the genes through changing the RF binding factor or other type of factors here. So there are more things we can do here, but I would rather go now back to my presentation to summarize what we want to show. Actually, let me open the presentation now. So you remember this, of course, interferon receptor is found to be very important here, and it's not a secret. So interferon is it's already a lot of studies done and showing that interferon signaling is very important for COVID infection, and actually for the severity of the COVID infection. But what we found also that for histone deacetylase, in fact, also here found as a master regulator. And why is that? And if you look at the literature, you can see that histone deacetylase is involved in many inflammatory processes. And it's already trials going on with these histone deacetylase inhibitors, and they are affecting many inflammatory processes, and specifically those lines which are related with the interleukin 1 and actually pro-inflammatory interleukin like interleukin 6 and interleukin 1 and other interleukins and TNF alpha. If you visit my previous seminar about analysis of expression data from COVID patient, you may remember that we found those receptors for those interleukins are playing important role in the severe affected patients. So here again, we see that this master regulator may play a really important role here. And to summarize, here we can focus, for instance, on that one SNP. Interestingly, that if you look at that SNP, then we can, of course, look at the databases and check how frequent is this SNP in the populations. So the SNP is actually changing. So the reference allele is T, and the alternative allele, which leads to the creation of IRF1 site, is C. And if you look at those African Americans, European population, and in general population as well, you can clearly see that the reference allele is more frequent. So with a high priority in African American, and also in British, it's 0.7, and the alternative allele only 0.24. But interestingly enough, that this alternative allele is becoming very frequent is in Chinese population. So you see here, C is actually 0.61 compared to 0.4. So that's may reflect more susceptibility in Chinese population. But that's, of course, a big speculation, but just a fact which you can observe here. With that, I want to finish with my presentation, and I'm open for the questions.
Thank you very much, Alexander, for your very interesting presentation. And now please put your questions in this Q&A block. You can please put your questions if you have any to any parts of the presentation. There is a question, report Genome Enhancer in table 4 about TF gain and loss. Maybe you can comment on this.
Okay.
A little bit on this table. In this table, do we have these IDs, SNP IDs?
Let me check. Let me open it.
This table, and maybe you just put.
Okay. So table 4, this one. So what was the question again?
Table 4 with gain and loss information. Is it a way to correlate to SNP IDs?
All right. Yes. So first of all, we can have a look at the full table. So, well, basically, this is a table, just statistics of gain and loss. To see the sites itself and the SNPs in them, basically, you have to look at the other files here. Let me check models. So we have gained and lost sites. Okay. Perfect. So that's right. So we have such a table. It's actually a big one, so it shows all the SNPs and all the sites which are affected. So you see this is a genomic position. This is the SNP ID, also saying which nucleotide is changing to what. And here you see the logo of that binding site, of that binding matrix. And then this is a particular region which is affected. Basically, you see G is changed to A, and it means so that this position 3 is now changed to A. As a result, you can, it is a gain of that site, and the score of that site at the reference genome is 0, so there is no site, because in this position is you see G, which doesn't fit to this consensus at all. And when A is staying here, the score of that site is actually 1. So it's a jump from 0 to 1. It's a very high jump. And you see other factors. You can actually filter this table by transcription factors. So we are interested, for instance, in IRF factors. So IRF then it's filtering this table by the area of factors. Let me check. Yes, so we see here all these factors, all these positions, and the RSs for them. And sometimes you see the factor, different position weight matrices are actually playing a role. So, and because there is a different variety of those matrices for different RF factors, and some of them are in different orientations. So this is a plus, this is minus.
Yeah. Okay, thank you. Basically, we see clearly that SNP IDs are given here,
Yes.
and alternative and reference alleles are also shown here. So the next question, which is more general one, maybe needs your comments. What kind of tissue was used for study of SNPs?
Well, I guess it was a blood because it's it's a genotyping. So the blood samples are taken from the subjects, and then they are used to run through the I guess through the microarrays, so Affymetric microarrays, which are checking a particular set of SNPs, and it's genotype each SNP for that particular patient. So it's we are talking about the generative the genome, so which is usually taken from blood.
Mm-hmm. And do you know what genome regions were searched actually in association with SNP? Was this
in our analysis? So in this study of that site on the COVID-19, so that's done actually genome-wide. It's a genome-wide association study. So all these SNPs are located across the whole genome, and then each SNP was correlated with a particular thing, like for instance with very severe respiratory confirmed COVID cases, or with hospitalized COVID cases. So it's a genome-wide. So, but when we do our analysis, then we focus on each SNP, first of all, just at the place where SNP is happening and this nucleotide position. But then we look around at the region around that SNP, as I said, 1,000 nucleotide around. So 500 left, 500 right from the SNP.
Mm-hmm. Okay. How could the distance between different TFBS change gene expression? And how this could be integrated from SNP data to drug development?
Okay, very good question. Thank you very much. So basically, as you can see here, the many SNPs which were found to be associated with this disease, they are colocalized. You see, these are the SNPs, and all of them are associated with COVID, and they happen to be in that regions altogether. It doesn't mean that all these subjects are carrying all those SNPs. Some are carrying one SNP, others are carrying another SNP. Maybe a few of them carrying several SNPs. But interesting that each of that SNP contribute to the binding of various transcription factors. And when those SNPs are located nearby, then they may influence several factors together. For instance, these two SNPs, they can influence these two transcription factor binding sites. That's why our model of composite modules, when several transcription factors binding to the regions and cooperate to each other, they may be affected exactly by those SNPs. And the second part of your question, how this can be used for the drug development, exactly that our report is answering. So by analyzing those SNPs and analyzing how they are influencing on the combination of transcription factors, we at the end detect the potential mechanism, and we find those master regulators which are actually affecting big part of the mechanism. And now these master regulators are considered as potential drug targets. And at the end of our report, we actually report what kind of drugs can be used against those drug targets. First of all, drugs which are known already for these types of diseases, like in this case, you see this PEG interferon or interferon beta is already known as a drug, also used in COVID. And in case of other targets like this, deacetylase, then other potential drugs are recommended here by using actually chemoinformatic software paths, which may can detect how drug potentially can inhibit a set of targets which are detected here.
Mm-hmm. Thank you. The next question. How many SNPs in spike protein are identified for SARS till now?
In spike proteins.
Protein, yeah, maybe you know this.
Yes, that's a good question. In fact, I was looking at the ACE2.
Yes, also it is a question about ACE2.
Yeah, I was looking at ACE2. In this particular study, ACE2 was not in the list. With ACE2 is a very complicated story, actually. So I haven't looked generally through all these GWAS studies so far, but just briefly, in those two studies I was analyzing, ACE2 was not no SNPs which was associated with ACE2 was detected as associated with COVID-19. That's
maybe more research will be done for sure.
Yeah, more research will be done. Exactly. Yeah.
Mm-hmm. Okay, thank you. A question about composite elements. I did not understand how composite modules or motif enrichment analysis around putative regulatory SNPs is performed.
Mm-hmm.
I mean, there is information of SNP changing the binding site of a specific TF. How can this be used to deduce other TF that interact to build a composite module?
Yeah, very good question. So in different ways, basically we can look at this in this example. So what happened here? So if we zoom out a little bit. So what happened here? You see those SNPs which are associated with COVID. They are here. And here are the genes. So we see here this interferon receptor gene, and by the way, nearby, and I missed, I don't have time now, there's another gene which is seems to be also involved, interleukin 10 receptor, and it's just nearby. And some of those SNPs are actually affecting, although they are located inside IRF receptor, but in fact, through the EQTL information, we know that they're affecting the NET gene. But first of all, talking about that. So the algorithm, what it's doing, it's first of all taking all those SNPs, and then it tries to find regions where those SNPs has to be analyzed. So here we see these regions. Let me just drag and drop them. These regulatory regions. If you drag them here, I will probably take out some of the information. This should be okay. So you see, I zoom in a little bit. So the algorithm goes through all these SNPs, and then it finds a region in genome, which is it has the most number of SNPs in that small region. Actually, this region is around 1,000 nucleotides. And it focuses on that region. So it takes this region, and it takes all those regions. So basically, if you open the regions as a table, we can see that we took 73 regions in different genes, Some of them are, some of them covering just one SNP, some of them covering several SNPs, but they are located either inside the gene or in 5' or 3' region of the gene. And they are covering the SNP, right? So we have all these regions. Now we run the composite module analysis just on the 3 regions and can compare them with other random regions in genome. And as a result, we get those composite modules. And just to demonstrate it a little bit more, let me again open this browser and take the found site. So here we have a extract. So we can look a bit more in detail. So the genetic algorithm finds the colocalization of these transcription factor binding sites near these SNPs. You see, it found that these SOX9, what I showed, OCT1, NANOC. So this combination of factors, they are frequently found around those SNPs which we analyze. So we basically, again, we take the SNP, we took a region around those SNPs, and we look with genetic algorithm what other transcription factors in combinations are frequently found in those regions.
Mm-hmm. All right. And the question, so additional question was about theory of this. Definitely we do have algorithm published, right? About
yes. Right, exactly.
So all the formulas are available and algorithm about this is published.
So this algorithm is called composite module analyst. And there's a paper, I think some years ago, which we published, which we benchmarked that algorithm. And now we actually improved it a little bit, and there's a certain advances in the latest time. But general basic algorithm is described and published.
Mm-hmm. Okay, thank you.
Mm-hmm.
The next question, all this analysis, I mean the workflow, could be done in plants to analyze SNPs in TFBS?
Mm-hmm.
Or so we focus solely on humans, or can be parts of this analysis done in plants?
Yeah, very good question. So this pipeline which I started, this Genome Enhancer, is solely for human data. So it's just human. But when we in the platform, then several other species can be used, although not the full pipeline, but only TRANSFAC parts of it. And we are talking about fruit fly, Drosophila, human mouse, rat, nematode, Saccharomyces cerevisiae, zebrafish. So we have these genomes of these organisms so far. So as for the plants, in TRANSFAC, there are plant matrices, and actually TRANSFAC is one of the, I would say, the very unique in general, but in terms of plants, it's really unique. So there is quite a big collection of plant factors and plant matrices are present in TRANSFAC. And I'm just looking in vertebrates. Maybe it's not
there
here.
Was just above, there was this rice, oryza.
Yeah.
And
we have those matrices.
Arabidopsis profile.
Exactly. Yes, and basically we can apply them to the sequences which you can load in the system as they are. You can load faster files with any plant species, and then apply the algorithms of those faster files. So in case of those genomes which are presented here, which I told, like Drosophila and Saccharomyces, then we have already implemented genomes, so you can load actually gene lists. But in case of plants, you don't have genome in the system, so but you still can load the sequence as in the form of FASTQ files, FASTA files.
Mm-hmm. Thank you. The next question is about TRANSFAC versus Jasper.
Okay. A very good question, and it's a frequently asked questions. Basically, as I mentioned, the difference is quite big. TRANSFAC collects all available, most of the available information about the transcription factor profiles from literature, from other databases. So basically, we look through all available databases like Jasper, Hokomoko, and other databases, and we re-evaluate those matrices and include them into TRANSFAC. In terms of numbers, as you remember, I showed that in TRANSFAC is around 7,600 positional weight matrices for vertebrates. If you look to Jasper, it's a bit more than 1,000. So definitely we have a lot, much more. Yeah. So, but this amount of matrices which we collect actually plays sometimes important role. As I showed, for instance, when you analyze data, it's not clear which kind of matrix which play for the same factor will play the role. So sometimes it's one matrix for a factor, for sometimes another one. So this amount of matrices which we collect in TRANSFAC is really important to analyze data in depth.
Thank you. In addition to the number of matrices itself, we can mention that, of course, TRANSFAC contains a lot of additional information about transcription factors. There are so-called reports on transcription factors where you can get a lot of additional information about function and structure of these transcription factors, because after you have identified putative binding sites, you're definitely interested to know more about these transcription factors, and TRANSFAC is very, very helpful with this information.
Yes, right. So
the next question is, could we extract the frequency matrix of transcription factor motif?
Yes, as soon as you have a license for the database access, so the subscription, and many universities actually have this, so you just have to ask in your library if you have a subscription to the database. So there are various ways of getting these matrices. So, for instance, if we looking at the database itself, there's interface. You can search for the particular matrix or particular transcription factor, and you can see the matrix here on the screen. Also, you can do it inside the platform. So if we have the matrix, as I showed you, for instance, if we have any site here like this one, basically you see the logo. You don't have here the matrix, but in the TRANSFAC database itself, here you can see the matrix. You see these values of the matrix here on the screen. So you can see each matrix separate when you have access to the database. Also, there's an option to have a subscription for the download version of the database, and in such case, you can download the whole library as a flat file with all the content.
All right, thank you very much. It seems that we answered all questions which are now in Q&A section. We have done with this, and we are very thankful for very productive participation, for your very interesting questions and comments. And we are looking forward to meet you at our next webinars, which we actually consider to talk more about transcription factors themselves. Just follow our information about webinars, and we will meet next times. Now we wish you a very good time of the day on your side. Goodbye.
Thank you. Goodbye.
Thank you very much. Goodbye.